Hi friends, it's Mr. Rogers here, and today we're going to go over the study guide. And really with the study guide, I'm going to give you guys some answers, and I'm hoping that these answers obviously help you kind of understand the study guide, but I kind of want to go through each one and make sure that we understand our thinking here. Understand, because I'm not going to ask the same questions on a test. The important things with study guides is not worry so much about the exact answers, but how we arrive to the answer. Like, why does that answer matter? So the first part of the study guide is actually these quizzes. You can take these quizzes. You can take them as much as you want. They'll give you answers back when you take them, except the water properties quiz, the top one. That one was more short answer, but the quiz or the test, sorry, is all multiple choice. So these other ones will probably be good ones to try. So number one, identify the organelle that breaks down dead and dying organelles into reusable proteins, lipids, and carbohydrates. The answer there is lysosomes. Lysosomes is actually one of my favorite organelles. It breaks down almost every single kind of object that a cell could eat or bring into itself or that it makes itself. And that makes the lysosomes super important, especially for animal cells, because we are constantly reusing parts. Now, lysosomes can also be dangerous. You have this organelle inside of you that could be popped at any point, and it could hurt your actual cell. It can even eat your cell from the inside out. And so lysosomes are super important little balloons of destruction. If they get popped, your cell could die. And so they're super important to keep safe. Your cell, make sure that it has a lot of protections for it. Some cells even have some extra things on the outside of lysosomes to kind of, if something bad happens, to kind of stop that from happening. But for the most part, these lysosomes are used to break down old things and to reuse the parts. List all the characteristics of the nucleus. Well, nucleuses are found in eukaryotes. They're found in the center of the cell. They contain DNA, and they're major parts of the control center of the cell. They're what's telling the cell what to do. All those would be great things to have down. List all the characteristics of the ribosome. It's found in all cells. Every single cell in this world has a ribosome. Its main function is to make proteins, which is super important because that's what a job of a cell is to make proteins. The other major things about ribosomes you could bring up is that they can be by themselves or they can be connected to the rough ER. I think they look like little hamburgers, but some people draw them as just little dots, but I think they look like hamburgers. There's a bottom bun and a top bun. But either way, those are some good things to put down. Explain the difference between the rough ER and smooth ER. The rough ER is got ribosomes attached to it, and it's going to make and help with the production of proteins, and it's the internal membrane system, internal highway membrane system of the cell. So it's going to help move things around these proteins so they get to the places they need to go faster. The smooth ER does not have any ribosomes, and its main job is to make lipids for the cell membrane. Explain the appearance and function of the Golgi apparatus. The Golgi apparatus is the mailroom of the cell. It's going to package up the proteins in the Lego box, put a stamp on it, put all the addresses on it, and send it on its way. So it's kind of like a post office of the cell. Compare and contrast prokaryotes and eukaryotes. This is something we did a last test. I just want to make sure that we understand the differences. Prokaryotes are bacteria. They are things with no nucleus. They have no membrane-bound organelles, meaning they have no complex organelles whatsoever. And eukaryotes are the opposite. They're basically other living things, so plants, animals, fungi. They have a nucleus. They have membrane-bound organelles, things like mitochondria, stuff like that. And they're more complex. That's a way to think about them. Prokaryotes came first. They're simple. Eukaryotes are more complex. Identify what organelle is only found in animal cells. That would be a centriole. A centriole is used for cellular division. We really don't know if it's actually used for cellular division. I brought this up in notes, but we've seen it be used there. Even though if we take it out, cell division still happens perfectly fine. It's still interesting that, it's used in that situation. Name a few other differences between plants and animal cells. Plants have a central vacuole. They have one vacuole. Animal cells we call vesicles, many vesicles. Plant cells have a cell wall, which is a rigid structure on the outside of a plant, outside of a cell, I should say, and it helps keep the structure of a cell. They also have chloroplasts, which are used for photosynthesis and for energy production. They typically, plant cells have less lysosomes, but I'm not really going to ask that, but it's just good to know. They have less lysosomes than animal cells. Those are the major differences. And animal cells have, obviously, that centriole. Identify which organelle is the powerhouse of the cell. What is its function? It is going to be the mitochondria, and its main job is to make energy for the cell. List the 3 parts of the cell theory. So we have all cells come from existing cells. So no cells just randomly appear in our universe. Cells are the most basic unit of function of life. So it's the smallest thing that we can classify as life. And all living things are made up of cells. So those 3 are the most important things. I've been known on a test to put some false answers on this kind of question, meaning like I'll give something that is technically true, but it's not part of the cell theory. So make sure you guys read through the cell theory question quite well. Identify what organelle can be used as storage. That is a vacuole or vesicle, depending if you're a plant or animal. They're exactly the same. It's just name differences. It's like how one country might name the same thing, something different than we do. So plants call it vacuole. We call it vesicle in animals. Identify what organelle produces ribosomes, and then the dark spot in the nucleus. It's called a nucleolus. That guy is going to be making the ribosomes. Explain the role of the cytoskeleton. The role of cytoskeleton is to help support and give shape and structure to the cell. It can also be used for transport, so things can move on it, kind of like roads. Cytoskeletons can also be used. You can see pictures of it down here. We have cilia. They say pili here, but it's technically cilia, and a flagellum. Those are technically made up of cytoskeleton. Eukaryote or prokaryote? Well, if you take a look here, it's kind of small. It says nucleus, so eukaryotes have a nucleus. This is a prokaryote because it doesn't have a nucleus. The DNA is free-floating and it doesn't have a bunch of other organelles. Explain endosymbiosis. Endosymbiosis theory is the idea that mitochondria and chloroplasts used to be their own living thing. We know this because we can't make mitochondria and chloroplasts. They can only make themselves, so we don't have the DNA to make them. They also have their own DNA inside of them. They have their own ribosomes inside of them that are unique. They have their own cell membrane, which is unique. And there are signs that they are truly their own living thing inside of your cell. So that's what the endosymbiosis theory is stating, that these 2 things, the mitochondria and chloroplast, mitochondria in animals and the mitochondria and chloroplast in plants, used to be their own living thing, and they were taken basically hostage or as a partner inside of a cell, and they still remain there to today. Let's start all the steps of the protein production. Well, we go from the nucleus and the DNA to the ribosome. So you can say rough ER if you want, the Golgi apparatus, to a vesicle or vacuole to the outside. It's a good way of putting it. I think that's good. We studied water the first week in this unit. I want you to draw water in your question spot 1. Make sure you put the charges in. So H2O. So there's 2 H's. I can probably go here. Give me a second here. There's 2 H's, and there's an H that looks like this. It's connected to an O, connected to an H. The H's are positive. The O is negative because the oxygen is a bully, and he holds on to all the extra electrons that he's supposed to be sharing with hydrogen, but he refuses. And so this is going to be negative. This is going to be positive. Explain why water is considered polar. What does polar mean? What is nonpolar? So water is polar because it has a charge. That's what polarity means, is that you have 2 opposite ends of a charge. So that we have a negative charge here, positive here. So this is technically a positive end over here, negative end over here, makes it polar. If you're nonpolar, it means you have 0 charge. You have no charge throughout your cell, or your molecule, sorry. And you don't want to interact with polar things, and polar things only want to interact with other polar things. So they kind of separate themselves out. If you put water droplets between 2 glass microscope slides, they stick together. Draw a picture of the water in between 2 microscope slides. Why is the glass now glued together? What about the water creates that? This is called adhesion. What is a clever way to remember that? So it just answered it for you. This is adhesion when 2 glass slides stick together. It's because glass has a charge and water has a charge, so they're both polar. So water likes sticking to it. That's called adhesion. I like to think of adhesive glue. You might have heard the word adhesive before. And so water acts as an adhesive between the 2 glass pieces. So adhesion is when water connects to something else besides itself. Cohesion, the next question, is when water sticks to itself. So on the Penny lab, water kept sticking onto itself and making a bigger and bigger bubble. This bigger and bigger bubble obviously got to a point where eventually the pressures of gravity was too much and it broke apart. Now the bonds between them are called hydrogen bonds. That's when other oxygens and hydrogens attract other guys that aren't actually part of their molecule. So for example, I'll have like a negative oxygen here connected to some H's here. And there's attraction. This attraction between this guy and this guy is called a hydrogen bond. Lake effect snow. Draw a picture of lake effect snow. What is the weather temperature right now compared to the air? What about in the summer? How does this affect the weather? So in the winter, the lake is warmer than the air, and so it's going to give off moisture, and it creates clouds and snowstorms and things like that. During the summer, the lake is colder than the air, and so therefore it cools off the area around the lake to be colder than the area further away from the lake. We learned about proteins, lipids, carbohydrates. Fill out the chart on your paper about each one. So the monomer for carbs is glucose. Use in living things, well, it's used as energy in living things. It's made up of C, H, and O, carbon, hydrogen, and oxygen, in equal amounts, basically, for the carbon and oxygen. So, for example, glucose is C6H12O6. So you want your carbons and your oxygens to be the same. Example, we have glucose, starch, fructose, cellulose. Anything that ends in /OSE is a sugar. Polar and nonpolar, carbs are very polar. I mean, their name is literally carbon hydrate, carbohydrate, so it's carbon water. Water is polar. Lipids, we never gave a monomer to lipids, so you don't have to worry about that. That's just X'd out. If you Googled it, they'll say something like triglyceride or something like that, but we don't have to worry about that. Use in living things, it's stored energy. Also can be used as cushion insulation in humans. It can be used as cell messengers or messengers in the body as a steroid. It can also be used like a wax, the waterproof things. So there's a bunch of different uses. What are they made up of? C, H, and O. Again, just like carbohydrates, except lipids have a ton of carbons and very few oxygens. Examples, we have saturated fats, unsaturated fats. We have steroids, which have 4 rings. Phospholipids, which have a polar end, but 2 very non-polar ends attached to it. And lipids are non-polar. That's kind of the distinguishing factor about lipids. They're non-polar. Proteins, monomers, amino acids. There's 20 of them. Each one is slightly different due to their R group. Their use in living things is basically they build everything. They're the building blocks of life. It's easier to say what they don't do than it is what they do. Made up of what atoms? They have carbon, hydrogen, nitrogen, and oxygen. So nitrogen is the addition here. So if you see something with a nitrogen in it, most likely it's a protein. Examples, tons and tons of examples. I mean, your muscles, the hemoglobin inside your red blood cells, a bunch of things. It's basically everything is made of protein. It's just different ways of making the same thing with different amount of amino acids. And proteins can be polar or nonpolar, depending on the R groups found in the actual structure. Explain endosymbiosis theory, mitochondrial chloroplast. We already kind of did it up top, so I'm not too worried about that. We already created a Venn diagram. I'm not too worried about that. Taxol is a cancer drug that breaks down cytoskeleton in cancerous cells. What effect could they have on the cell? Well, we know that cytoskeleton kind of builds and gives structure to a cell, so maybe the cell will fall apart. We also know that it's part of transportation, so maybe things won't be able to move as well. We also know cytoskeleton can be the flagellum and the cilia of a cell, so maybe they can't move altogether the cell. So a bunch of different things you could put. Cyanide pops the membrane surrounding the lysosome. What does that do to the cell? It basically kills the cell. The things in the lysosome can break down anything, so eat the cell away. Chromate is a chemical that makes mitochondria work extra hard for a bit, then it dies. What are the short-term and long-term effects? Can we make more mitochondria? So short-term effects, if the mitochondria is working harder, you're going to get more energy. Long-term effects, if it dies, you're going to stop getting energy from it. And can we make more mitochondria? We can't. Mitochondria have to make other mitochondria. And so once they die in a cell, they're dead. They're gone. Steroids send a signal to the cell to make more proteins. What organelle would be doing extra work, and what organelle would you need to make more of said ribosome? Well, that kind of gives away the first answer. What organelle would be doing most of the work? It's ribosome. That makes proteins. And so if I'm making more proteins, that's what's going to be doing the extra work. And what organelle would you need to make more of the said ribosomes? That would be the nucleolus. Cactuses contain giant vacuoles in their cells. Why does that make sense? What would be in the vacuole? Well, it would be holding water. Cactuses obviously live in dry places. So their vacuoles are going to be really, really big for the time when they do get water. They can store as much water as possible and take out what they need as they go along. Write out the 3 cell theory statements. We already did that. Saturated fats, unsaturated fats are different. Explain how. Saturated fats are solids at room temperatures. They are from animals. They do not have any double bonds. They only have single bonds. Unsaturated fats have double bonds, so that means they have bends in their legs, if you want to say that. They have they're liquid at room temperature, and they're from plants. Coconut oil is from a plant, so this should be an unsaturated fat. Steak is from an animal, so it's a saturated fat. So I kind of went through everything. Obviously, we have these. We have the questions from the quizzes. The test, I believe, is a very fair test. It is only about 20 questions long. You have 25 minutes. Please make sure we don't cheat. I'm going to have Go Guardian on. So if you get caught cheating, you get a 0, and there's no coming back from that. You get a 0. There's no retakes, nothing. And so that's not a good start. A good way to start your next quarter. So please make sure you're being honest. I'd rather have you struggle and not do well. And so then we can learn from your mistakes and retake it than lie yourself through the test, because you're going to get caught. I swear to God, I'm going to catch you cheat. So please don't cheat. Please do it honestly. Good luck on your study guide. If you need any help, just let me know, and I'll talk to you guys soon.